营养健康所毕业的博士啊，然后在 Harvard Stem Cell Institute receive her postdoc training， 然后他是2014年回国，然后他现在的工作的主要是做肝脏方面的工作，但今天他给我们带来的故事是跟尿酸代谢有关的大家欢迎。
Good afternoon. So thanks very much for the opportunity. So I'm going to share a molecule that we are currently working on. It's uric acid. So I'm sure all of you are familiar with uric acid. So the left is the chemical structure of this molecule. So it is actually a degradation product of purine degradation in the liver. And the most of the uric acid is excreted through kidney. And also a portion, about 1/3 of the uric acid is excreted through intestine. So there is actually a significant amount of uric acid in the gut lumen. So the reason uric acid is popular is probably because of the high incidence of hyperuricemia. So in China and worldwide, in China, about 13% of the population has been diagnosed with hyperuricemia. And it is well known that this hyperuricemia has been associated with increased risk of many diseases, including cardiovascular disease, kidney disease, or obesity and metabolic disorders. And to be noted that low uric acid levels in the patient with low uric acid levels due to genetic effects or drug treatment, they also have increased risk of diseases such as neuronal degenerative diseases, for example, Parkinson's disease and Alzheimer's disease. So when we search the literature, it's actually the biological function of uric acid, especially soluble uric acid, is understudied. So we basically have not much idea about the pathological role or the physiological role of uric acid. And from an evolutionary angle, that human beings is actually specific mammals that have very specific high level of uric acid. As you can see here, that in the mouse, the common model we're using to do experiment, the uric level is around 30 micromolar. But in humans, we actually usually have about 300 micromolar. It's about 10 times the fold of the uric acid levels in the mouse. And the reason is that during evolution, we actually lose the gene encoding for uricase, which metabolize uric acid to anatomy in most mammals. But evolutionarily, that the gene encoding uricase becomes a pseudogene in humans, resulting in high uric acid levels in human beings. So there exists an intriguing question is about what is exactly the significance of the evolved high uric acid levels in human beings. So as previously mentioned, that high uric acids are associated with obesity. And then the evolutionary, we have developed a high uric acid level status. And some evolutionists predict that or prospect that the reason that the high uric acid level actually confer us a protective effect in the environment with energy depletion. You know that during evolution, we actually usually face a challenge of food leakage. So we are curious about whether uric acid is a contributing factor to our fat or energy storage in humans or to the development of obesity obesity. So we first look at the human data. We try to search for human data that tell us some clues. Actually, there are not much data doing research about how uric acid affects obesity development. Usually those population studies usually focused on whether if we lose weight that we can reduce uric acid. However, we did notice 3 studies. So one of the studies that they found that the serum uric acid concentration can predict a subsequent weight gain in 5 years. Basically, they measured the uric acid levels in people, and then they track the weight gain in 5 years. What they discovered is that if you have higher uric acid levels at the beginning, you are more likely to have more weight gain after 5 years. In the other 2 studies is about they monitored the body weight after uric acid lowering therapies. And what they noticed that is after 3 months of uric acid lowering therapy treatment, the patient can have a reduction of 2 kilograms in body weight. So this suggests that uric acid may be a contributor to our fat storage or obese development. So in order to, we wanted to see whether uric acid and obesity actually have a causality link. To do this, we started from genome association study, searching data from genome-wide association studies. So what we do is that we searched for genetic variants that in the transcription level, in the transcription region of genes, and then identified those genetic variants that they are both associated with uric acid levels and obesity traits such as body mass index. So in total, we identified 26 genes. So among which we are most particularly interested in the gene TIP60. The reason is the TIP60 encodes for lysine acetyltransferase 5. So it's a well-known acetyltransferase that's responsible for the acetylysine of histone or non-histone proteins. Previously, we discovered MRG15, a chromatin remodeler in this complex that play an important role in the metabolic diseases. So we started with TIP60. So by carefully looking at the genetic variants in the TIP60 transcriptional region, that we noticed that those variants are closely, in the genetic level, are closely associated with serum uric acid and body mass index, and also some other metabolic traits such as triglyceride, cholesterol, and type 2 diabetes. So we first, as I just mentioned, that uric acid is most produced in the liver. So we first knock out TIP60 in the liver cells. And very remarkably that we observed a very significant decrease of the liver uric acid levels as soon as 1 week after TIP60 depletion. So consistent with the genetic clue showing that TIP60 is closely associated with human uric acid levels. In TIP60, that is actually he's a chromatin, he's an enzyme that's responsible for the histone acetylation. So we further discovered that it actually can directly bind to a series of genes that in the purine metabolism, for example, MPD2 and NT5E-XDH, such as, so all of them showed a consistent downregulation after TIP60 depletion. And then we see a decreased chromatin accessibility and reduced histone acetylation near these genes after TIP60 depletion. And then what we found very interestingly, that's also consistent with human genetic data, these mice are very resistant to high-fat diet-induced obesity. As you can see here, we can see a very clear reduction in body weight after high-fat diet fading in these mice with liver TYP60 depletion. And this change in body weight only happens when the mice are fed with high-fat diet. And then whether this body weight reduction is associated with uric acid. So we fed these mice with uric acid. And what we found is that if we reverse the uric acid levels to its original level, and the body weight change has been reversed, so that the TYP60 depletion caused body weight reduction in high-fat diet can be reversed by the uric acid feeding. And we also confirmed this in different models. One of them is we also knocked out several genes that in uric acid synthesis that previously I mentioned. All of them are involved in purine degradation. And then after we knock out these 3 genes in the liver, we also see a similar reduction in body weight upon high-fat diet feeding. And this can also be recapitulated by the treatment of the uric acid synthesis inhibitor, the triple-stat, that we can also see a downregulation of this body weight in high-fat diet. So this suggests that uric acid, at least in the mouse model, actually contribute to high-fat diet-induced obesity. So what caused the body weight reduction in these animals? So what we found first is that we didn't see any change in food intake or energy consumption. As you probably already guessed from the title, that what we found is that this uric acid, the reduced uric acid caused reduced intestinal dietary lipid absorption. So here you can see that we can see increased fecal free fatty acids, cholesterol, and then also some transcriptional signatures about in the intestine, for example, CD36, which is a fatty acid transporter. And this can also be confirmed, and this change can also be reversed if we feed the animals with uric acid. And this can also be confirmed in different animal models, for example, in the treatment of the uric acid synthesis inhibitor, that we also observed an increased the remaining free fatty acid in the feces from these animals, and also downregulation of CD36 in the intestine. So what we confirm from here is that uric acid can regulate the intestinal lipid absorption. We also wanted to find whether in humans that we can also see a similar effect. So we did studies from 2 different cohorts. So this one is published. Oh, sorry, I'm not sure. Okay. So this is a published data set who did lipidomic study from humans with normal uric acid levels and hyperuricemia. And what we analyzed the lipid content from lipid compositions from this patient, from this population. And what we found that uric acid is very significantly associated with dietary lipid levels. For example, what I highlight here is DHA, linoleic acid. So those those fatty acids are essential fatty acids that our body cannot really produce it. We can also obtain this fatty acid from diet. So this suggests that uric acid is associated with the take-up or absorption of the fatty acids from the diet. And we also collaborated with Changzhou, the second hospital in Changzhou, that we collected feces samples from around 100 people. And then in these samples, we checked the fecal lipid levels in this around 100 human samples. And what we found is that the serum uric acid is significantly negatively associated with fecal lipid levels, suggesting that the one with high uric acid actually has less free fatty acid remaining in the feces, suggesting that uric acid actually promotes the absorption of free fatty acid. And then how uric acid modulates the dietary lipid absorption, and our further study suggests that this is through modulating gut microbiota. We did this by different approaches. For example, the antibiotic drinking in different mouse models, and the transplantation of the fecal microbiota transplantation, suggesting that this effect is mostly through modulating gut microbiota. And then we also identified a bacterial species is called lactogenesin bacteria. So this L-genesin abundance is affected by uric acid levels. And I just mentioned, because 1/3 of the uric acid is excreted in intestine. So there is a significant amount of uric acid in the gut lumen. So we detected the uric acid concentrations in the gut lumen and see a very significant association with the abundance with L-genesin. For example, that if you have decreased, the animal models have decreased uric acid levels, you usually have a significantly higher abundance of this bacteria. And then in vitro, we can see as low as 10 micromolar treatment of uric acid. Uric acid can have a very significant inhibitory role to the growth of this L-genesin bacteria. And then we also see that the L-genesin also responsible for most of the phenotypes we see in the animals. After transplantation, we can see a reduced lipid absorption and body weight gain after high-fat diet. And we also identified metabolites such as PLA that's responsible, that derived from L-genesin, that suppressed lipid absorption in the intestine. A short summary of this story is that I think most important part is through this story we realized that uric acid actually have some functions that we were not aware before. That what we found is that uric acid can modulate the composition of gut microbiota, which can suppress some lipid transporter gene expression in the intestine and resulting in affected dietary lipid absorption. Of course, we also identified a master regulator of these genes in purine, master regulators of purine metabolic genes in targeting TYP60. For example, we also encapsulated lipid nanoparticles to target TYP60 that in the obese mouse models, and we can see a very dramatic decrease in uric acid levels and the protection from high-fat diet-induced obesity in the mouse. And then I think this is the only start that we need to understand how uric acid actually functions in our body. That there are a lot of hypotheses actually from the evolutionists, because that we have developed in evolution to have a very high level of uric acid. What exactly the evolutionary significance of increased uric acid levels in humans? There are different hypotheses. For example, it will regulate the metabolism that I just mentioned. It will promote the fat storage, the energy storage, and it probably will. It is also important. It is also prospected to have important roles in maintaining the blood pressure, and it may also have immune regulation roles, and also maybe regulate longevity and cognition. Speaking of longevity, I actually add this slide because this conference is more about aging. Actually, that people already done a study in a mouse model that increased uric acid levels causes that significant increased lifespan in female. But we don't exactly know the underlying mechanisms yet. And then when you search the literature, a lot of reviews will tell you how uric acid works. And the primary function of uric acid, they consider is uric acid is an antioxidant. So it is. They actually calculated the antioxidant capacity of uric acid, and what they said is that because we have around above 300 micromolar uric acid in our blood, so they think they calculated and consider like 60% of the antioxidant capacity is contributed by uric acid in our blood. So this is a very high amount of contribution for antioxidant capacity of uric acid, although I don't know how they do the calculation. But I believe that, but does uric acid only function as a simple antioxidant? I think the answer is no. That when we search the literature, that we noticed a study that done by David Saptini in 2000 and published in 2017. What they did is that they simply culture a tumor cell line with a human plasma-like medium. And they found that compared with the tumor cells we cultured in a normal culture medium, that those cells have dramatically remodeled the pyrimidine metabolism. And what they've identified uric acid as the endogenous inhibitor of UMPS, which is a UMP synthesis, and found that and they also discovered that those tumor cells in this medium can influence, and uric acid can actually influence the sensibility to the 5-fluorouracil. So this actually, I don't know, this paper is published in 2017, but after that, we're still not aware that when we do cultural work or animal or mouse model studies, that we basically neglect the existence of the species difference in the uric acid levels. And it turned out these uric acid levels may affect the tumor cell growth or a lot of other things we probably are not aware at this time. But how to study uric acid? We currently don't have a nice mouse model to study uric acid. The people try to knock out uricase and to get hyperuricemia mouse models. But in this mouse model, these leaders, because they usually have the mouse model with double knockout of uricase have very high levels of uric acid. And 60% of this mouse will die in embryo because it's very toxic to kidney or other tissues. So most of the leaders will die. And then we notice a publication that not only we don't have uricase in human beings, we lose uricase in evolution, but also we have quite a difference in XOR expression. XOR is the enzyme that converts dennin to uric acid. And here is a paper published in 2000. And then you can notice here that the human liver, the XOR expression is much lower than the mouse XOR. So suggesting that in humans, the synthesis of uric acid is actually slowered. So to do that, we replaced the mouse XOR with a human homolog. So with this mouse, we get a humanized mouse that weighs around, at the baseline, about 160 micromolar of uric acid. And these mouse are totally healthy after birth. So basically, you can breed them like normal animal. And what we found that from this, we did some preliminary study in this mouse model. What we found that actually these uric acid levels is quite responsible to nutrient status. For example, if we fast this mouse like overnight, you can see a double increase, a twofold increase of uric acid levels. And these uric acid levels can be reversed after around like 6 hours feeding. So, but in wild-type mouse, you basically see no change in uric acid levels. So with this mouse, and in humans, actually, some report also observed the increased uric acid levels in humans after fasting. So, this actually tells us uric acid is actually dynamically regulated during feeding or fasting or other stresses that we basically couldn't see in the wild-type mouse, but probably we can study in this humanized mouse. And what we did is that we also collected to observe a systemic change of these animals, that we collected tissues from this mouse, different tissues from this mouse, after feeding or fasting. And from intestine, as you can clearly see here, that we see a much increased expression of a lot of lipid transport genes in this humanized mouse. So suggesting that the increased uric acid or the humanized uric acid model actually have a more sensitive response to fasting. The increases after fasting, it actually activates the lipid absorption programs in the intestine. And in addition to intestine, we also collected different tissues. And what we observed is that we also see very remarkably alteration of gene transcription in hypothalamus and adipose tissues and also immune cells. So I think those are also interesting information that we probably will focus on next time, focus on in the future. Hopefully we can have some more data next time. So take-home message is that we found that uric acid may have some important functions. What we actually previously ignored, that when we do research, that the first signal is that we found UA can actually modulate the composition of gut microbiota and regulating the dietary lipid absorption, at least in humans. And then we have generated mouse models with humanized uric acid synthesis process that can facilitate our research on uric acid levels, uric acid function. That's all about my talk. So this work is primarily done by Tianzhen and Qi Yunfei, and also our collaborators in different institutes. Thank you very much. I'm happy to take any questions.
Actually, a very interesting story. I have two questions. The first one is, what is the effect of the uric acid on the TIP60 activity? Is it possible during the hyper uric acid and TIP60 activity will be changed? The first one. The second one is, what is the TIP60 phenotype in terms of the fatty liver left fold?
The first question we don't know. So what we have found now is TIP60 actually can regulate the UA synthesis in the liver, but we don't know in a different way whether uric acid can actually affect the TIP60
maybe for
activity.
the
I think
patients
that's
with hyper uric acid, the TIP60 is a decreased expression or something, right?
Yes, we don't know, but it's a good point that we probably need to look at in the future. But in the humanized mouse, when we do fed fasting and we compare the transcriptional changes in different tissues, we actually didn't see much difference in liver tissue. So we don't know yet.
Reduced body weight very significantly.
No,
and the liver
the
fat
humanized mouse with high uric acid levels in the mouse. And the second question is, yes, TIP60 knockout, because it has a very dramatic reduction in body weight. So it has improved metabolic sensitivity to glucose or insulin, and also decrease fatty liver.
Thank you. Very interesting.
Any questions? I have a question for you, Chiro. Have you checked the GLP-1 levels where you change the uric acid levels?
No.
Because after you.
It will not affect. We don't see changes in food intake, and we don't see in this mouse model. We don't see. At least at the basal level, we don't see change in body weight or dramatic change in metabolic features. But we don't. But we probably will see after like high-fat diet challenging.
Okay, thank you. Okay.